So hopefully, is it in slide viewer now? It could just, okay, great. So thank you everyone for having me here today. I'm so excited to be a part of this group and to talk to you a little bit about some of the dissertation research that I did. And so I put this presentation together just to sort of like introduce myself, give you some highlights, give you some background on some of the other things that are going on in the project. So with that, I graduated, so I finished my PhD in agronomy from K-State University this December. Prior to that, I did a master's in natural resource management up at the University of Alaska Fairbanks, where I primarily focused on managed grazing and and nutrient cycling in the subarctic. And then before that, I got a bachelor's degree in economics and a bachelor's degree in ecology. And so some of my work experience, I worked as a graduate assistant in the environmental quality lab at K-State University, where I really focused on soil health metrics. So all of the ones that are, you know, you would be familiar with out of Cornell. I ran lots of those samples. We did some water sampling. I looked at different ways to measure organic nutrients, soil respiration, active carbon, microbial biomass, carbon, nitrogen, phosphorus. So all of those types of measurements. In Alaska, I worked at the Large Animal Research Station, and I worked primarily with musk oxen, caribou, and reindeer. You can see them in the picture to the right. So I was in charge of their managed grazing, and we did a lot of managed grazing trials to suppress weeds. We did some work with musk oxen and electric fence and some of those. And then I looked at some on-farm economics as well. And then prior to that, prior to having a family, I was a warrant officer in the United States Army. So I'm going to focus today on some of the work I did at Kansas State. Kansas in general has a really difficult problem with water quality. Most of their watersheds are adversely impacted by phosphorus runoff, and this can be a really costly problem. Some research has estimated the eutrophication of waters to cost up to upwards of $2.2 billion per year. And in Kansas, you know, it's all fresh water, so it really takes a hit from some of that phosphorus runoff. And that picture at the far left is of a reservoir that's about 10 minutes from campus. And you can see all that light green is an algal bloom that happened in 2015. And so, you know, there was a lot of pressure to start doing some research at the field scale to look at different ways that we could mitigate some of these issues. And so the PI on the project, Dr. Nathan Nelson, and my advisor, Dr. Peter Tomlinson, we're looking at different options. And one of the things that comes up a lot is conservation management. And so when I'm thinking about conservation management, I'm primarily talking about cover cropping and no-till to reduce the amount of nutrients that are lost from fields, so in-field practices. And some of the things that are really well supported is that, you know, no-till and cover crops can improve soil health. They can reduce the amount of sediment loss, and they can reduce nitrate leaching. And so, you know, but there was a lot of questions about what the role of soil health was in nutrient loss and what the ultimate impacts of that were on water quality. And so one of the things that was really missing was that the impact of some of this conservation management is unclear from a phosphorus standpoint. And to the right, I just grabbed some different journal articles that sort of document some of these problems with some of the conservation practices and their potential impact on phosphorus loss. And then the other thing that was identified is there was very little research in regards to conservation management, how they interact together. So if you have multiple different types, you know, if you're doing cover cropping and no-till, and then how we incorporate fertilizer management into that. And so that was the primary thrust of my work. Dr. Nelson, he set up, you know, they wanted to test this at the field scale. So this is the site that I worked on was very, very large. Each plot was 1.5 to 2 acres in size. So this was at a field scale. It was set up so the whole site was a no-till, and it was a corn soybean rotation. And so this type of setup is very common for our region of Kansas. And we were testing different combinations of cover crop or no cover crop and different types of fertilizer management. And what's really neat is that we were able to capture the runoff from each plot. And so the picture to the right is an aerial photo of the site. And we had a full factorial design. So we had different combinations, and each of those red triangles is one of these inset flumes. And so basically what it means is that each of the plots was hydrologically isolated. And so we were able to capture the runoff from each treatment and then make some really direct links between management and then what we were seeing in our runoff and our water quality. And just as a side note, this little yellow circle is a pickup truck. So it gives you a sense of the size of the site. And so these were our treatments. These were how they were laid out. We had a cover crop or no cover crop treatment. And that cover crop did differ year to year. We had triticale rapeseed mix or a rye 1 year. And so it's really difficult to say anything. That is one of the caveats of this work, is that we can't say anything specific about a cover crop treatment. But one of the things that the research team felt strongly about is that we wanted to do what farmers in our area were doing. And one of those things was making decisions about cover crop mix. And often that was what was the most cost effective. And so that was the way it was managed. And then we did 3 types of fertilizer management. So we had a control, which was no phosphorus. We had a spring injected ammonium polyphosphate and a fall broadcast diammonium phosphate, which were both applied at 27 kilos per hectare. And then we tested the soil. We tested the top 6 inches of soil at 2-inch increments because we wanted to get at some possible stratification that might be happening. And so this is a video right off the bat. This is something Dr. Nelson took. And so the video on the right is just to illustrate the impact on sediment loss. And so the video on the left, excuse me, is a plot that has no cover crop, and a video on the right is one that has a cover crop. And so this is the same rain event, and you can just see the immediate impact it had on how much sediment and water was coming with that. And so that was also borne out in the data. So the graph to the left is a graph showing some of our erosion measurements. So this is how much soil or sediment was in our runoff water. And you can see that there was significantly more sediment in plots that had no cover crop. And so this actually got at something that some of our farmers and producers wanted to know, is whether they needed a cover crop if they had it in no-till already. And so that was one of the things that is an outcome of this work, is that we can say, yes, it really, even in no-till, it still really reduced the amount of sediment lost. And so depending on the year and how much rain, but we averaged about 60–70% reduction. And this is really interesting. So the graph on the left is a hydrograph, and these are hydrographs from 2 plots, one with a cover crop, one with no cover crop. And so on the vertical axis is the flow rate. So it's basically how much water was coming off of that particular treatment. And on the horizontal axis is the timestamp. So basically, where this yellow line and green line is where the runoff started and then when it ended. And what's really interesting is, so the brown line is the treatment that did not have a cover crop, and you can see the intensity was really high. So that water peaked at a much higher rate, but it also finished sooner. And so the cover crop plot, its intensity was less, but the water stayed on that plot longer. And so the outcome of that is that the intensity was greater in the no cover crop plots, but the volume was the same. So it had no impact in our study on how much water came off of that plot, but it did change the dynamics. And then the phosphorus in the runoff. Surprisingly, even though we had read some other reports where phosphorus was increased from cover crops, we also saw that in our study. So this graph to the left is the total amount of phosphorus that we recorded in our runoff. And so you can see in 2016, 2017, there was no difference, but in 2018, we had more total phosphorus in our cover crop plots in our runoff than no cover crop. And then the graph to the right, where we have dissolved reactive P, we see that the cover crop treatment increased the amount of dissolved reactive P in each year. And so that research was courtesy of Dr. Nathan Nelson. He and his team were the ones that recorded this particular result. And so that really led to where my work came in, was this knowledge gap. And really, you know, a lot of people are asking why would we know that cover crops reduce sediment, and we know that sediment can be bound, or phosphorus can be bound to sediment. So why would we see an increase in some of these things? And so that's one of those questions that we really don't have a good answer for. But one of these things is, what we do know is that no-till frequently has problems with nutrient stratification. So if it's applied to the surface, there's no tillage, so there's no mixing. And another, you know, there's been research also shown that the cover crop residues have really labile forms of phosphorus. And so if there's residue on the surface and you have a freeze-thaw cycle, it could lyse those cells, and then that could be released into the water. But it still doesn't necessarily account for all of the phosphorus that we've been seeing. And so one of the things that I was interested in was how these management strategies interact with each other, and then how do they affect where the phosphorus is being stored. And then when we see increases in potentially soil health, how does that change the phosphorus cycling? And so that was our goal, was just to document these linkages, start investigating some of the mechanisms, but really so that we can make these optimal management recommendations to producers and stakeholders. Because one of the important things is that if we're recommending that they implement something that takes time and money and effort, we want to make sure that we're getting at what the goal is of their operation and what they're trying to achieve. And so some of our initial work, you know, we did have a lot of stratification, which wasn't surprising because we're in no-till, so there's no mixing. So we used Mehlich 3, which is an agronomically significant P measurement used in Kansas frequently. And we saw that even under all of the different treatments, we had higher phosphorus at the top 2 inches, and that incrementally went lower. And so I just want to orient you to this graph. It's a little bit flipped, and it sometimes takes a minute to look at it. But basically, this 0 here, this is the top of the surface of the soil. So that's where the soil surface is, and then the horizontal axis is really how much phosphorus there is. And so you can see at the shallower depths, we had more phosphorus than deeper down. And so when we looked at our stratification, we zeroed into that top layer, because that top layer of soil is really that's part that's going to be interacting with the precipitation. And so it was interesting because we saw a fertilizer effect, but really that effect was just that we had more fertilizer in our fertilized treatments than in our not fertilized treatments, but that the fall broadcast and spring injected was the same, which is partially expected. If you're not fertilizing something, you wouldn't expect it to have as high phosphorus levels, but also a little bit unusual considering that spring injected is injected below the surface. So it was an interesting path that sort of led us to ask some of our further on questions. And so, you know, Mehlich 3 is an agronomic measure. We decided to look at easily extracted phosphorus. So we used some very weak organic acids that are similar to what you would expect roots or microbes to exude to be able to make that phosphorus available. And we started looking to see if we could find some more nuance in that. And so what's interesting, this is a similar graph. So on the vertical axis, you can see that 0, that's the soil surface. And down here, the 15, that's the depth. So at 15 centimeters. And then these lines are our 6 different treatment types. So our 3 fertilizer treatments combined with cover crop or no cover crop. And so what was interesting is that in the no phosphorus plots, we didn't see any stratification. And then in the spring injected no cover crop, so if you had no cover crop, but just a spring injected fertilizer application, there was also no stratification. But in the fall broadcast and spring injected cover crop, we saw high stratification. So this is interesting, with the fall broadcast, that's not a very surprising result because we're applying phosphorus to the surface of the soil. And so it makes sense that we would have a high concentration. But with this spring injected, the big divide here is that addition of the cover crop really changed the dynamics in the spring injected application of fertilizer. And so we looked at how our soil health fits in here. And really the story with soil health and our treatments was that across the board, almost invariably, cover crops increased all of our soil health metrics. So we saw that the cover crops increased our total organic carbon by about 10 to 15%. We saw that it increased our microbial biomass carbon. And so microbial biomass carbon is a measure of how big that microbial community is. So it's quite a gross measure, but it is a good way to look at that overall size. And we did see a lot of seasonality in it. So we took samples in the fall and the spring. And so in the spring, following that cover crop, we saw that the cover crop had about 25% more microbial biomass carbon than if there was no cover crop. And one of the things that I want to keep your mind there is that this microbial biomass is really a measure of those cells. And so those cells have carbon, nitrogen, and phosphorus inside of them as well. And the reason this is of interest in a phosphorus sense is because those cells are very sensitive to lysing. So if you have wet dry periods or freeze thaw cycles, those cells can then basically just can be lysed and spill their guts. And so one of the things that's important is that if there's a lot of phosphorus in those cells, they could potentially be a significant source of phosphorus and then contribute that to water quality issues. And so that's exactly what we looked at, is this microbial biomass phosphorus. And I just want to lead you through it. So we have on the left-hand side how much microbial biomass phosphorus. So that's how much phosphorus is in the cell itself. And then our 6 different treatment types. And what I thought was really interesting is that within fall broadcast, it didn't matter if you had a cover crop or no cover crop. The amount of microbial biomass phosphorus was the same, statistically the same. And same if there was no fertilizer added, they were statistically the same, there was no difference. What's really interesting is that in the spring injected, so when we spring injected ammonium polyphosphate, we saw that there was significantly more microbial biomass phosphorus under cover crops than no cover crops. And so what does this all mean? How do we bring these things all together? Well, this is still a work in progress. And so I hesitate, it's really interesting, but it also hasn't been confirmed in other locations. And so I don't want to overinterpret what our results mean. But what our thinking is that a potential mechanism, there might be several things going on why cover crops increase the amount of dissolved reactive phosphorus. And some of our thoughts is that we just have water sitting on that plot longer. So it might be dissolving more phosphorus out of the soil and then contributing that to runoff. It might be stratification dynamics. So potentially, if that cover crop in the spring injected is maybe moving some of that phosphorus out of that subsurface placement and putting it at the surface, leading to higher stratification, that also could contribute to phosphorus and runoff. And then also, if we have just better soil health, higher microbial activity, they could be cycling nutrients more quickly. But we really need some more details to flesh out some of those things. But we're thinking that those are potentially ways that we're seeing what we're seeing in our runoff results. And that's some of the next steps, is that these biological measures are going to, or these results are going to be connected to more biological measures. So we have some PLFA data that we're just now running. We look at enzyme stoichiometry. So enzymes are expressed by microbes and plants in response to nutrient status in the soil. And so we're going to be looking at some of those to see if we can see if maybe microbes are responding to those nutrient dynamics. And then we're also going to look at our crop and cover crop phosphorus uptake and deposition, and how that is playing into everything. And then, like I mentioned, it's really important to confirm results in a different location, in different times, different years, different crops to make sure that this is something and not just a one-off thing. And another thing that I like to bring up too, when we're talking about cover crops and phosphorus in water, it may not mean that cover crops are bad. It may mean that we need to do more research in adjusting our fertilizer recommendations or fertilizer application in response to cover crops so that we can optimize some of the benefits we get from cover crops and also minimize some of these other unintended consequences. And so the takeaway from all this, cover crops reduced erosion more than no-till alone. And I feel comfortable, this is really well supported in the literature. This isn't gonna blow anyone away, but it really, we saw some significant differences. Again, our soil health improved with our cover crops, but that potential increase of cycling, like that's something that's a big question mark and depends on the year, and is a lot of assumptions about how those nutrients are turning over. And then one of the things that we need to investigate further is how cover crops are then changing phosphorus cycling, and in particular in relationship to spring injected phosphorus management. And it's important that this be confirmed in other studies, because if spring injected phosphorus is one of our best management practices and cover crops are one of our best management practices, but we're suggesting they be put together, then we need to make sure that we're not negating what we're trying to achieve. And so I know that was a bit of a whirlwind. This was a huge project with a lot of people working on it, and the work of my advisor, Dr. Tomlinson, the PI of the project, Dr. Nelson, my committee member, Dr. Presley, and Catherine Stewart and Elliot Carver, other graduate students. I just want to acknowledge all the hard work that they put in. And so with that, I would be happy to answer any questions, or you can email me if you have something offline that you wanted to talk about.